So today we've got our lecture looking at our proteins. So proteins are made up of individual units called amino acids. So the first part of the lecture will be focusing on these building blocks, which are our amino acids. So just like the nucleotides made up our nucleic acids, which were our DNA and RNA, the amino acids are connected together to make our proteins. So we can say that the amino acids are the building blocks of the proteins because individual amino acids are actually connected together. And when we have these individual amino acids connected together, that's what makes our proteins. We'll also have a look at some of the essential amino acids. So these are the amino acids that must be obtained from diet. So our body is actually unable to make these, so we have to get them from the diet. There are also what we call non-essential amino acids, which our body can actually make, and we can also get these from the diet as well. So we'll have a look at some of the properties of amino acids. So we'll see that the properties of amino acids are actually specified by their R group, which is the variable group within the amino acid. And we'll also look at the way amino acids can actually function as a buffer. So part of the amino acid can actually act as an acid, and the other part can act as a base. And this allows it to form what we're going to have a look at called the Zwitter ion. So the Zwitter ion is where we have charged states within the amino acid. We'll also have a brief look at cysteine. So we've seen what cysteine can do in earlier lectures where we actually have two of these sulfhydryl groups, which is the R group within our cysteine, and we lose the hydrogens, and that allows us to connect the sulfurs together through what we call a disulfide bond. We'll also have a little bit of a look at our important small peptides. So a peptide is basically just a very small protein, so less than 20 amino acids. We define it as a peptide rather than a protein. We'll then have a look at some of the different types of protein structure that actually make up our proteins. So we'll see that the structure of a protein is very much going to dictate how it functions. So, for example, it's just like any object. We create our objects in a certain way so they're going to be able to perform a particular function. For example, with a drink bottle, you need a hollow container that can hold water and then dispense the water. The structure of the drink bottle dictates what you can do with it. You can drink out of it. So it's much the same as our proteins. They have a certain structural arrangement which gives them their function. For example, hemoglobin has a particular structural arrangement that is different from every other protein, and that gives it a particular function. Its structure, arranged in this way, actually allows it to function to carry oxygen around the body, whereas a different protein will be structured differently and will have another function. So we'll have a look at this relationship between structure and function throughout the session. So protein hydrolysis is actually our way of breaking down our proteins, so we can break them into individual amino acids. So by adding water to the protein, we're actually able to break down the protein. So we do actually require heat and also acid to do this. So mostly this happens under laboratory conditions. So the next thing after that we'll have a look at is our protein denaturation. Basically, denaturation just means removal of structure. So we're removing the protein structure. So I showed you on the right hand side here that the protein structure is very important for how it functions. So if we remove the structure, not only do we remove all the structural integrity, but we also remove the function. For example, if we go back to our drink bottle example, then if we melt our drink bottle down, destroy its structure, it can no longer function as a drink bottle. So just like with a protein, if you destroy its structure, it no longer functions as it did before. And then the last thing we'll have a look at is some of the different classes of our proteins in terms of structural proteins like collagen and keratin, protective proteins like antibodies, and we'll have also look at some other types like membrane proteins and catalytic enzymes, which are enzymes that drive the chemical reactions. These are our enzymes. Alright, so we get proteins into the body through our diet. So we're able to obtain these from animal products like different meats and other sources like nuts and other bean-like substances will have some protein in it. So the idea here is that we need to actually break down our protein into the individual amino acids so we can absorb them into the body and then use these proteins that we've taken in through the diet to make up other important proteins that we need in the body. For example, we need the collagen protein to maintain our skin. So you see here that the overall mass of the cell is also very high in protein, so 15%. So we need to be having a constant supply of proteins to be making more proteins, which helps sustain us. Alright, so we're going to be referring a lot to the structure of the amino acid. So all amino acids have the same structure. The only part that ever changes is what we show here as the side chain. So the side chain is our variable group. And we represent this as R, because remember, R can represent a lot of different things. So we consider this side chain separately to the rest of the structure. So the side chain is what changes, and then the rest of it actually never changes. What we've got down the bottom here is what we call the backbone atoms. So the backbone atoms are going to be identical no matter what amino acid you look in. They're all going to have these same groups. So they're going to have two functional groups. One of these functional groups is the amino group, also known as the amine group. They're also going to have a carboxyl group, which is actually our carboxylic acid group. So we saw these functional groups in our organic chemistry lecture. And we also have a single hydrogen attached. So an amino acid is made up of these four groups. We've got the variable R group, the two functional groups, and the hydrogen. So which of these four groups that is attached to our central carbon is a part of the side chain? So the side chain is our variable region, designated as R in this picture. So remember, R can represent different groups of atoms. So we'll see some of the different side chains that the amino acids can actually have on the coming slides. What about the backbone of the amino acid? What groups of atoms make up the backbone? So it's actually the combination of the amino group, the central carbon, the carboxyl group, and also the hydrogen. So these are the backbone atoms that do not change. So no matter what amino acid we look at, they're still going to have these different groups. So they're going to have the two functional groups, the hydrogen and the central carbon, which we call the alpha carbon. So we can call the alpha carbon the central carbon also. Alright, so we're going to focus on these 20 standard amino acids that we naturally find in our proteins. So of these amino acids, they're all different because they have these different R groups. Remember, the R group is the part of the amino acid that changes from one to the next. For example, alanine has a different R group to glycine. So depending on what this R group is, that is what determines the actual properties shown by that amino acid. So we don't look at the backbone atoms to work out properties. So we see that the properties actually come from our R group. So the R group can specify that the amino acid is going to be non-polar if it's just carbons and hydrogens connected by non-polar covalent bonds, whereas if it's polar and neutral, then there's going to be at least some oxygens and nitrogens, and this will mean some of the bonds within that structure are now polar. If they're a acidic group, they're actually going to contain a carboxylic acid group, and that carboxylic acid group can act like an acid, so give away its H+. And if it's a basic group, it's actually going to contain a NH2, which can gain H+. So we'll see the consequence of this on the coming slides. So we can separate our amino acids into one of these four groups. They're either going to be non-polar, made up of just carbons and hydrogens, polar neutral, so they're uncharged, but they'll contain our oxygen and or nitrogen. And if they're acidic, then we're going to have the carboxylic acid group, and if they're basic, they're going to contain our amine group here. So these two at the bottom here, acidic and basic, have the possibility of becoming charged. So if they are acidic, they can actually become negatively charged, and the basic ones can become positively charged. So I'll show you why this is in the coming slides. Alright, so we've got a few examples here of our amino acids. So we can see in the grey here is our backbone. So the backbone is the part of the amino acid that never changes. So they've all got the amino group, the carboxylic acid group, and the hydrogen, as well as our central carbon, which connects to 4 different groups of atoms, which actually makes this chiral. So one way we can designate chirality is basically by just putting a star there. So we saw that in one of the earlier lectures. Alright, but what we're focusing on for this slide is what is in this colour up top here. So we've got this group protruding upwards. So these groups are our R groups. So remember back to the earlier slide, we saw that variable R group. So this variable R group is different depending on what amino acid we have. So alanine has a different R group to serine, which has a different R group to aspartic acid, etc. So let's have a look at what makes each of these groups either polar, non-polar, or another property. So our alanine is actually non-polar. Why? Because it contains only carbons and hydrogens. And this means it's what we call hydrophobic. Hydrophobic is what non-polar compounds are because they hate water. So they don't want to be near water. What about if it's polar? How is a polar compound different? So here we see it has oxygen. Could also be nitrogen in another example. Because it contains oxygen, this actually makes it polar. So instead of being water-hating, it's now what we call hydrophilic, which is water-loving. So if it contains oxygens or nitrogens in the side chain, then the amino acid is happy to be in water, whereas if it contains only carbons and hydrogens, it hates water and is what we call hydrophobic. So we've got an example of our basic amino acid here, which is lysine. So you can see that it has our amine group up the top here, and the amine group actually has basic characteristics, so it can accept H+ to become NH3+, and that will be meaning it's now positively charged. What about our acidic amino acid? What functional group does that have? It actually has our carboxylic acid functional group, which can give away H+. And when the carboxylic acid gives away H+, it's now negatively charged. We'll also have a look at our cysteine here, which is the only amino acid R group that contains our thiol group. So remember that the thiol group is SH, and we can have two of these thiol groups coming together to form a disulfide bond. Alright, so on the left-hand side here, we can see the amino acids grouped into these different categories that we've defined. So the nonpolar amino acids are those that have side chains. Remember, this is our R group, the side chain. And that side chain, if it's nonpolar, is going to be made up of exclusively carbons and hydrogens. So if we look at all of these structures, they all have carbons and hydrogens on their side chain, where we have the green here, which is indicating the side chain. So tryptophan and methionine actually do have one sulfur and one nitrogen respectively, but because there's many more carbons and hydrogens, we still class these as being nonpolar. Compare this to where we have our polar side chains. Many more of our atoms are now either oxygens or nitrogens, which means they contain these polar covalent bonds, and they're happy to be in water. So you can see these groups in pink here actually have a higher proportion of oxygens, nitrogens, or sulfurs compared to what we saw above here, which now makes them water-loving, hydrophilic, and this means they are polar. So down the bottom here we have our acidic and basic amino acids. So two of our amino acids have the possibility of performing as acids. So we can see they both contain our carboxyl group. So remember, the carboxyl group was one of our functional groups within the backbone, but if we actually have this carboxylic acid group in the as a part of the R group, this actually makes us acidic as an amino acid. It's now an acidic amino acid, and what do acids do? They donate H+. So both aspartate and glutamate, also known as aspartic acid and glutamic acid, are actually able to donate H+. And then on the right hand side here, these are our functional groups that contain the amine. So we've got quite a few nitrogens within these functional groups, and these can act as bases, which means they are going to be accepting H+. So remember what I said in the introduction about our amino acids? So they fall into either the essential group or what we call the non-essential. So if something is essential, this means that it must come from diet, because the body actually cannot make it. Compare this to where we have a non-essential amino acid. This can actually come from either the diet or the body can make it itself as well. So key difference here is that we need to obtain the essential amino acid from the diet, whereas the non-essential amino acid can be made by the body and also obtained from the diet. So which part of our amino acid do we look at to determine whether it is polar or non-polar? So we look at our variable R region and then identify whether it has carbons and hydrogens only, that'll make it non-polar, or if it has a higher proportion of things like oxygen, nitrogen, or sulfur, which will make it polar. And remember, if it contains an acidic or basic group, this will also mean that the amino acid is actually polar, but it actually has the ability to become charged now once it gains or loses the H+. So what makes an amino acid essential? We call an essential amino acid one that must be obtained from the diet because the body cannot make it itself. How is this different from a non-essential amino acid? Well, non-essential amino acids can actually be made by the body directly, and we can also obtain them from the diet, so it's a bit easier for us to obtain those amino acids.